So the first part of chapter 5 is a little bit of review. So we talked about energy and how we need energy in order to maintain homeostasis, right? To maintain our body. Things tend to go to a state of disorder. So you have to put energy into it to keep it ordered, right? So to maintain order, we need energy. The problem, however, comes from the fact that when we convert from one form of energy to another, it is inefficient. We don't lose or gain energy. There's still the same amount of energy, so there's a conservation of it. But some of the energy is lost as heat. Okay? So think about when you exercise. You're converting chemical energy into mechanical energy. So you're converting sugar into muscle movement, and you get hot. You sweat, right? You have to get rid of the excess heat by sweating. So we lose that energy as heat. And so that is a core source of energy because it's really hard to get it back and use it to do work, right? So generally we just lose that heat energy to the environment. So we have energy moving in the ecosystem. So we have plants that are capable of capturing light energy, right? And then they create chemical energy that then the animals are able to eat. And then we can use that energy, we could also eat other animals, but we can use that energy, but eventually we decompose, and then that material that is broken down, there's also a release of heat. So the idea here is that we have energy conversions, and we lose some energy in the form of heat, okay? Okay, so when we look at metabolism, remember that we defined metabolism already, and so this is all of the chemical reactions that occur in your body. So what are the chemical reactions that build stuff? What are those called? Anabolic, okay? So you probably have heard or seen that word previously because anabolic is sometimes used in terms of anabolic steroids, right? So when you think about anabolic steroids, those cause you to build muscle, right? So they're anabolic. So anabolic is building of molecules. Now, is anabolic typically endergonic or exergonic? Endergonic, which means that it requires energy to build molecules. So we can store energy in those molecular bonds, okay? So what is the opposite of anabolic? It is catabolic. And it is exergonic. Okay, so it releases energy. So we're going to define another form of energy besides heat. And we're going to call it free energy. And this is energy that is available to do work. And the symbol that is used to describe the amount of free energy is G. So G is the symbol. So like in a mathematical equation, G would mean free energy. So we have a value that is the difference in G. So this little triangle is delta, right? Right. So if you're into sororities or fraternities, that's delta symbol, right? So delta G is the difference between the amount of energy that is used and the amount of energy that is produced, okay? So this is equal to the difference between the amount of energy used and the amount stored. Okay. So we're not going to look at specific values for G, but we're going to talk about whether it's a positive delta G or a negative delta G. So if it is a positive delta G, that means that the amount that is stored is greater than the amount that is used, right? So this is that we have stored energy, right? Some chemical reactions require energy, but they also produce energy. So this means that it produced energy, it stored it, we can use that energy, that's free energy that we can use for work, okay? So which one of these do you think is positive delta G up here? Anabolic. So this is a positive delta G, right? So we're storing energy. Catabolic would be a negative delta G, right? So that means that when we broke it down, right, we are releasing energy, and so there's less energy that we've stored, right? Okay. So when we look at the chemical reactions in our body, right, we can see that anabolic, anabolic stores free energy, so it's a positive. Catabolic releases energy, so it's a negative, okay? So it's releasing energy into the environment. Energy released. Yep, that's right. Okay. I heard rumblings, and so I started to doubt myself, but I checked my notes. That's correct. Okay. Okay. Now, this is where it gets very interesting, because now we're going to start talking about the energy that is in the atoms and in the electrons. Okay. So this is a review, but we'll go back over it. We have the nucleus of my atom. We have these different orbits that go around. Okay. How many electrons can the first orbit store? 2. How many can the second and the third? 8. This is the octet rule. Okay. Now, if we have an electron and it goes from a low energy and it moves up to this higher orbit, is that storing energy or releasing energy? Storing, right? So it is storing because we're pulling it away. So if you think about 2 magnets and you pull them away, we're storing energy, and when we release them, the electron will move back down, okay? So we're storing energy. So this is storing. If it goes the opposite direction, like if it started way out here and then it moved in, that is releasing energy. Okay, so that's how we can store and release at the atomic level. And when we have atoms sharing electrons, sometimes the electrons get pulled away from the nucleus and energy is stored. And then when we break the bond, it comes back in and energy is released. Okay, so we can store energy in chemical bonds. So we have a special type of chemical reactions that are called oxidation reduction chemical reactions. And we're going to, I'm going to give you the mnemonic device to learn, which is called oil rig. This does not mean anything, has nothing to do with oil rigs, right? It's just a way to remember it, okay? So what this means, oil is, this means oxidation is losing. Okay. So we could ask, what is it losing? There's a couple of things it can lose, right? It could lose electrons, right? So if we have electrons leaving the chemical formula, it is losing them. right? So that substance, when it goes through a chemical reaction and it's lost electrons, that means it has been oxidized, okay? The other thing that it can lose is hydrogen atoms. So I'll just put a hydrogen there, a hydrogen atom. So anytime when you see something losing a hydrogen atom, that means that it is being oxidized, okay? Now the other thing that's kind of weird about this is it also has to do, when we're talking about electrons, it can lose electron or the electron could move away, okay? So the electron could move away from the nucleus. And then it is oxidized. Okay. So let's look at an example of oxidation first. So oxidation would be like this. What type of molecule is that? Does anybody know? That is glucose. If we combine it with 6 oxygen, we can then convert this, and I'm going to put an arrow in this way. I'm going to put another arrow back this way in a second. We can convert this to CO2 plus H2O plus ATP, and it's actually 6 CO2 plus 6 H2O, sorry. So the reason why I put this 6 in here is that I'm trying to account for all the carbon. So I'm trying to balance my chemical reaction here. So if we look at here, there's 6 carbon atoms in glucose. So there's got to be 6 carbon atoms on the other side of the chemical reaction because we can't lose material. It's got to be accounted for in some way. So notice that there's 6 carbon, okay? So then if we look at oxygen, there's 6 oxygen here. And then how many here? 12, right? Because there's 6 oxygen. So 6 and 12 are 18. So then we have 12 here, and we have 6 here. So there's 18 on each side. And the same thing with hydrogen, okay? Now you notice that what has happened here is that carbon, right, has essentially lost its hydrogen atoms, right? So the hydrogen has gone away from the carbon, right? It happens to be in the presence of oxygen, so oxidizing makes sense because it's oxygen, right? So this is what is referred to as oxidation. Okay, it's gone, and we produced energy. But we've released the energy from the chemical bonds that used to be present between the carbon and the hydrogen. So glucose has lost hydrogen, and it has become oxidized. So we'll say the glucose was oxidized, okay? Okay, so let's look at the opposite of oxidation, which is Rig. So what do you think that means? Reduction is gaining, right? What's it's gaining? It's gaining electrons. It's gaining hydrogen atoms. And it is hugging the electrons closer to the nucleus. Wait, yeah. Reduction is gaining. Okay, so it's gaining electrons. Okay. Okay. So this has another name to it, and so it is called cellular respiration. Okay. Okay. So this chemical reaction can occur in the opposite direction. And what is that called when you combine carbon dioxide with water and you produce sugar? We can't do it, but plants can. Photosynthesis. And so this is reduction. and it is photosynthesis. Okay. So breaking it down, catabolic would be negative G, so there'd be a negative G here, right? And there would be a positive G here, right? So we're endergonic. So this would be exergonic. This would be endergonic because we're taking and we're storing energy. So we're adding hydrogen to the carbon, and so therefore it is reduced because reduction is gain. Right? So let's look at another example. So there is a chemical in your body that is called NADH. And this is a chemical like ATP. So it's, I think the long name is something like nicotinamine, adenine, something like that. Anyway, it's got a long name that goes with it, but it's like ATP, so it stores energy. And it can go from this to this. Oops, NAD, I meant to do big A. Okay, so that should read NAD. Oops, NAD. This is a really important molecule because we can store energy in it, and it's kind of interesting. They're now selling supplements. So you could actually buy supplements of NADH. I don't know if there's much evidence that suggests that it actually gives you more energy, but supposedly as we age, we have less of it, and so it might be that might be a cause for aging, and so it's kind of considered to be currently the fountain of youth. You can get this. Okay, so the chemical reaction going in the to the right, is that oxidation or reduction? Oxidation. Because we're losing the hydrogen, right? And then this would be reduction. Let's see if I have another example. Okay, we can take iron and we can combine it with oxygen. And then this produces, in one direction, ferrous oxide, which is Fe2O3. Okay. What do you think ferrous oxide is commonly called? Rust, right? So this is rust. Okay. So when we look at this, the oxygen is present. Notice that there's no oxygen up here. So you can't rely upon oxygen being present to determine whether it's oxidation or reduction. But iron right here is hugging its electrons in really close, right? And so here it's having to share its electrons with the oxygen. And so it has actually lost some of its electrons. So the electrons have actually moved away from its nucleus because now it has to share them. So oxygen is losing. The iron lost some of its electrons. They moved further out onto their shell. And so that would be an example of oxidation too. We don't really know how to take it the other way, right? You can't take something that's rust and rebuild it. Right? Once your car starts to rust, that's pretty much you have to do something really dramatic, right? Okay. Okay, so this is just an image. You don't need to memorize this. This is actually from the internet. So this just shows that NAD positive, right? And then if it gains 2 hydrogens, then it can be converted. And so that would be reduction because it's gaining. When it loses the hydrogen atoms, it loses. So it is oxidized. Okay, so it just takes a little while to sit down and think about this. Okay, and so we're going to, before we get back to that oxidation reduction, we're going to take a little break. And we're talking about enzymes, because we've already talked about enzymes quite a bit. Okay, So enzymes are interesting because they catalyze chemical reactions. Oh, I can't spell catalyze today. Catalyze, I think that's right. Chemical reactions, which means that they speed them up. They do this by binding to the substrate and breaking it apart. So enzymes have a substrate binding surface or a substrate binding site. Oops, not site. So the substrate binding site is usually visualized as a lock. And then the substrate binds and it fits into that lock like a key. So this is the lock, right? So that's my lock. And the substrate fits like a key. So this binding site is due to the three-dimensional folding of the protein. So the protein folds into, it's a globular protein, so it folds into a complex 3D shape. And so that's what creates that binding site. So if we look at a diagram from your book, right? The gray thing, which this could be a good quiz question if I took away all the pictures or all the labels. This is actually the enzyme, okay? It's a globular protein. It has a complex 3D shape. This is the substrate binding site. Notice that what binds to it is the substrate. And notice in this particular instance is that it's breaking it apart, right? Some enzymes build, some enzymes are anabolic. This would be a catabolic. So it's breaking it apart and it's releasing the products, okay? So the important thing to realize is that enzymes do not get used up. So they do not change. So they can release and then they can bind some new substrate and release them again. What did we find out in the lab about what can damage the structure of an enzyme? Temperature, right? So if we heated this up, the protein that is the enzyme will unfold and it will lose its ability to bind. And what is that unfolding called? Denaturation, right? So it denatures, it unfolds. Okay, so when we look at these enzymes, we have to talk about how we would regulate them, right? So I'm going to skip over that. So we're going to talk about how we regulate enzymes. Because we want enzymes to work sometimes, but not all the time, right? So sometimes we want the enzymes to be active, sometimes we don't want them to be active. And so we have what are referred to as inhibitors. And these are molecules that prevent the enzymes from working. So it keeps them from breaking down a substance. Now, sometimes these enzymes are competitive. So, for example, competitive inhibitors these bind to the same site as the substrate. So if the inhibitor is present, it's competing for access to the same site. And so the substrate can't bind there. And sometimes we use drugs. There's drugs that are used to do this. And one example of a drug, so this is an example, is a drug called statin. And does anybody, or statins, does anybody know What do you take statins for? High cholesterol, right? So this actually, these are chemicals that prevent the cholesterol, the enzyme that would convert lipids to cholesterol. So this prevents the enzyme from converting lipids to cholesterol, right? So that is an inhibitor. So you take that as a drug, okay? And statins will help you reduce your overall cholesterol level, okay? Sometimes the inhibitors are not competitive. So if we look at an example of an inhibitor that's not competitive, we can see this example. So ignore this this left side. So ignore this one for right now, okay? And let's look at the inhibition. So you'll notice here that the substrates, the site that the substrates would normally bind is present, but the inhibitor binds to the enzyme and it causes a change in the 3D shape. So this is called non-competitive inhibition, okay? So non-competitive is where the inhibitor binds to the enzyme causing a change in its shape. And so the substrate can no longer bind. So the substrate can't bind. Okay. So that's non-competitive inhibition. So now all that means is that the inhibitor is not binding at the same site as the substrate. So it changes its shape, and that is what is called allosteric. So the changing of the shape is called allosteric. So this would also be non-competitive, but it's also allosteric inhibition. Okay, one more type of inhibition, and this is what is referred to as feedback inhibition. And so this is where the product that is produced then acts to inhibit the enzyme. So if we're trying to regulate the amount of a substance in a cell, for example, we can have a set point, right? And if that substance goes over its set point, then it comes back around and it inhibits the chemical reaction from occurring, so you get kind of a homeostasis. So feedback inhibition can be visualized like this. Okay, so this is an example of a substrate binding, right? It binds, it breaks down, it produces it, and this is actually a series of enzymatic steps. So, but the end product that is produced by this chemical reaction, so this is more than one type of chemical reaction, would come back and it would inhibit the substrate from binding again. Okay? So that's what is referred to as feedback. So that is all inhibition, but if we go back to this diagram from your book, you'll see here that not only do we have inhibitors, but we also have activators. So activators will cause the substrate to bind. So sometimes the enzyme is not able to do its job unless an activator is present. So unless something binds to it to cause a change in the shape so that the substrate can bind. So some of these activators are kind of interesting because we call them coenzymes. Oops. So you write activators, coenzymes. And coenzymes typically are like vitamins. So vitamins oftentimes act as coenzymes. So the vitamin has to be present in order for the chemical reaction to occur, because it aids the enzyme in doing its work. So for example, vitamin C, if you have a lack of vitamin C, you can't produce collagen. And collagen is a protein that is in our skin, but also in our bones. And so you get what? What do you get if you don't have vitamin C? Scurvy, right? So the pirates, right, they had to make sure they had vitamin C or they became scurvy dogs, right? Okay, so the scurvy is where your skin starts to break down and you can't grow and your bones become weak, right, because you can't produce collagen. We also sometimes have, so this example would be vitamin C. We also have cofactors. And cofactors are specifically inorganic. Inorganic. So like iron is a cofactor and magnesium is a cofactor. So iron and magnesium are common cofactors that have to be present to help the enzyme do its work. Right? So iron has to be present, not just in like hemoglobin, but in other chemical reactions in your body, iron is necessary. Right? And those are inorganic because they don't have what? Carbon. Right? Vitamins are carbon-based or organic. Minerals are inorganic. Okay, so this leads us to the assignment. Okay, so this is another regulation, another way to regulate whether or not an enzyme is present. Okay, so what type of molecule is lactase? It is a protein. It is an enzyme. Right? Lactase is coded for by a gene. So a gene is a piece of DNA that codes for a protein. Okay, so we have a lactase gene. So the gene is DNA, but it codes for that enzyme. Okay. Now, associated with genes, specifically enzymes, a lot of the times there are genes that turn on or turn off the actual coding gene, and this is called a genetic switch. Okay. So this is a piece of DNA that can turn on or turn off the gene. Okay, so if it's turned on, lactase is being produced. If it's turned off, lactase is not being produced. Okay, so that's the genetic switch. So this helps to regulate when the enzyme will be produced and when it is not produced. Okay. So lactase persistence suggests that the gene is turned on. Now, is that common in adult mammals to have lactase turned on? No. So if you think about it, the veterinarian always says, don't feed your adult cats cow's milk or your dog's cow's milk, right? Because they'll make them sick. And that's because they'll take in the lactose, but they won't be able to digest it. So for some reason, and the video will talk about this, there seems to be certain situations in which lactase persistence in humans has evolved. And so there's some questions that relate to lactase persistence. And then they ask you to take from that information, they ask you to look at a different situation, which is the digestion of starch rather than the digestion of lactose. Okay, so that's the assignment. So I think that's pretty much all the background information that you need for that assignment to complete that assignment by next Tuesday. Okay. Okay. So I'll just introduce this. This is the next couple things that we'll be talking about. So when we talk about that cellular respiration, the first part of cellular respiration is called glycolysis. So this is the first part of cellular respiration. Okay. Glycolysis, the chemical reactions that occur during glycolysis are anaerobic. What does that mean? Don't need oxygen. It also, they occur in the cytoplasm instead of the mitochondria, so not in the mitochondria. And it does produce more energy than it uses, so it's got a positive delta G. Okay. So what this means is it produces more ATP than it uses. Oops, than it uses. Okay, the ending product of glycolysis is for every glucose, so we have one glucose molecule, we get 2 molecules of pyruvate. Okay, so pyruvate is the ending product of glycolysis. Now that pyruvate could then be used to produce, through fermentation, alcohol. So microorganisms like bacteria are capable of glycolysis. It can also be used in muscle cells to produce lactic acid. Or the pyruvate itself could then be oxidized in a chemical reaction that is referred to as the Krebs cycle, or what is called the citric acid cycle. So just when you're reading about this, you'll see a diagram that looks something, or some images that look like this, okay? We're not going to talk about this in a lot of detail, but notice that we start here with glucose. Notice ATP, is that reduction or oxidation when it goes to ADP? Is it losing electrons or gaining electrons? It's losing, right? So it's oxidation, right? And then we get these molecules that then get fed into another series of chemical reactions, right? And the end product is pyruvate. So when ADP goes to ATP, that's storing energy, right? And so that's what is referred to as reduction, okay? So this is the chemical reaction that occurs inside of your cytoplasm. And notice the enzymes, right? Anytime you see an ACE, all these enzymes are necessary. All these enzymes are coded for by genes. And so it's a series of chemical reactions that eventually lead you to the production of this. So pyruvate then could go in one or two directions. Okay, so let's just say we have pyruvate. So let's ignore fermentation, don't write that down. Pyruvate. This will be the last thing we'll talk about. So if oxygen is not present, it is converted into lactic acid or ethanol. So different bacteria, for example, if you're talking about, this would be fermentation, but if you're talking about if you're trying to produce beer by using a yeast that breaks down the sugars in malts to produce alcohol, this is the chemical reaction that would take place. If you have muscle cells that don't have oxygen in them, they would produce lactic acid as a byproduct. This is not very efficient, okay? If oxygen is present, we get what is called the citric acid cycle. And this occurs in the mitochondria. And it is very efficient. So it produces a lot of energy. Citric acid cycle. Okay. That is also called the Krebs cycle. So Krebs is with a K. This is called the Krebs cycle. So we're going to talk about this in a little bit more detail. So you might want to read ahead and look at the pictures. Don't panic over it. I'll tell you exactly what you need to know. Okay, so have a good rest of your week. Remember, there's no labs this week. And make sure that you do your assignments. And so I don't think so we're not going to have a quiz on Monday or next Tuesday. And I'm not going to do another lecture. So you don't need to watch a lecture or study for the quiz. So next Tuesday, we'll just continue with the lecture that I started.